Part 5. Human colour receptors. No two retinas are alike. In this image, we see colorized photographs of 5 different people's foveas. There are 2 things I'd like you to notice. First of all, the blue cones, depicted here in blue colour, are sparsely distributed across the fovea, whereas the red and green cones are far more numerous. The second thing I'd like you to notice is that the relative proportion of red to green cones varies enormously across individuals. As we'll see shortly, extreme differences in the proportion of red to green cones has implications for colour blindness. This graph shows the responsiveness of each of the 4 photoreceptors, the rods, the red, the green and the blue cones, to different wavelengths of light. As you can see, each photoreceptor type is optimally sensitive to a particular wavelength, and then this sensitivity falls off as the wavelength changes. That said, there's still an enormous amount of overlap in the various sensitivity profiles. This overlap is particularly pronounced in the case of red and green cones. Now, this is interesting because we often think of red and green as being in some way opposite of one another. Think back to your primary school art class with the colour wheel. Red is often seen as the opposite of green. But as you can see in this diagram, both red and green cones have very similar wavelength response profiles, which are in no sense opposite of each other. Clearly, something else is going on. The brain's response to wavelength is determined by the relative response of each of the 3 cone types. This implies that there are just 3 dimensions underlying our experience of colour. Because all wavelength spectra, no matter how complex they are reduced to just 3 dimensions by the human brain, we can't reliably predict the almost infinite number of possible spectra which might have driven them from the cone responses themselves. That is to say, the relationship between the spectral input and the cone responses is non-invertible. Here's an illustrative example. The spectrum at the top contains a relatively broad distribution of red and green light. If we look at the cones to the right, we see a strong response from the red and green cones and a negligible response from the blue cones. Now, let's look at the spectrum below. We have a very narrow band but intense stimulus peaking at 580 nanometers. Now, looking at the cone responses to its right, again, we see strong responses from the red and green cones and a negligible response from the blue cones. Whereas the 2 sets of stimuli are very different, the cone responses are identical. As we can see, we can't predict what the stimulus was based only on the cone responses. That's what we mean by non-invertible. We can predict the cone responses based on the stimulus, but not vice versa. Because the cone responses are identical in both cases, this implies that the 2 stimuli will actually appear identical. This is called a metamer, a situation when 2 physically distinct stimuli appear identical.